Hey, welcome back. Today we're taking a closer look at prokaryotes. I'm back with you live here. Hello. So today we're taking a closer look at prokaryotes, our second lesson in the cells unit, also known as bacteria. Prokaryotes, bacteria. Bacteria are prokaryotes. Most prokaryotes, almost all prokaryotes are bacteria. So we use those words interchangeably throughout. Prokaryotes, bacteria. Prokaryotes are divided into two domains. We discussed this yesterday: archaeobacteria and eubacteria. So archaeobacteria, they live in extreme environments, usually, which would be unusual today, but in primitive Earth. So back when Earth was a rock just flying around the sun, there would have been a lot of these archaic environments for the archaeobacteria to live in, like volcanoes or really acidic pits or very hot oceans. So for example, an example of an archaeobacteria is the thermacetophile, which lives in hot sulfur springs with temperatures as high as 80 degrees Celsius, which doesn't sound so hot until you realize we're talking about Celsius and not Fahrenheit. It'd be 176 Fahrenheit, scalding, and a pH as low as 2. Most of the environments we live in are pH between 6 and 8. So pH of 2 is a lot different than a pH of 6 to 8. And then there are eubacteria, which represent the more common forms of bacteria that live in places that you and I would live in. They live in the ground outside, they live in the water, they live even inside of organisms such as yourself. You have hundreds of bacteria, different species of bacteria living inside of you right now. So to better learn the parts of bacteria, we're going to take a look at this little guy here, just this generic picture of a bacteria, as we take a look at some of the parts. So all bacteria are single-celled. We discussed that yesterday. There are no multicellular bacteria. And that's in contrast to multicellular organisms such as yourself, which have about 30 trillion cells. So one cell versus 30 trillion, yeah, you're a lot more than a bacteria. They're generally very small, between 1 and 10 micrometers. That U looking there is a micro, Greek letter micro. It stands for 1/1,000,000th of a meter. So think of a meter stick, the yay long, there would be 1,000,000 of those bacteria lined up from one end to the other. Most bacteria have one circular strand of DNA, and that's right here, highlighted in the purple because it is purple colored, and one circular strand. We'll talk more about eukaryotic DNA, such as yourself, later in the year, and we'll find out that eukaryotes tend to have several strands that are linear, not circular. So I know that the picture here makes it look like a big mess and not circular at all, but really just think of a rubber band. And if a rubber band is circular, we all know that, but if you let it kind of roll up, it gets all tangled up and knotty, and that's what's going on with the bacterial DNA here. It is indeed just one circular strand of DNA. All right, next up, the cell membrane. We just spent a whole unit on the cell membrane. All cells have cell membranes. So bacteria, definitely a cell, definitely has a cell membrane. It is a phospholipid bilayer, just like all other membranes, and the purpose of it is to determine what gets into and what gets out of the cell. That's what cell membranes do. They keep the interior of the cell the interior, and they keep the exterior of the cell the exterior. Just like any other cell membrane, it is selectively permeable. Some things can go right through it, other things cannot. So it is in charge of what gets in or out of the cell. The plasma membrane really is the barrier of what is and what is outside of the cell. Next up, outside of the cell membrane, most bacteria also have a cell wall. So it's like the plant cell wall. In a lot of ways, it serves as a rigid structure. It doesn't allow the cell to get too big. So I think of osmosis the other day. A cell in a hypotonic environment will swell up and explode. Well, generally not bacteria cells. That cell wall is a lot firmer, a lot more solid of a structure, and it's not going to let that water balloon-like plasma membrane get too large and eventually burst. The cell wall is not selectively permeable. It's like Swiss cheese. It has holes in it everywhere. It is fully permeable. Anything can get right through it. The word we have for full of holes is porous. So we'll take a quick aside here, and while we're on bacterial cell walls, we'll take a quick look at how the drug penicillin works, because it works on the bacteria cell walls. So first of all, a huge difference between plant cell walls and bacteria cell walls is what they're made of. Bacteria cell walls are typically made of a polypeptide, see the peptide word in there, and polysaccharides, the glycan. So polypeptides and polysaccharides, we should start to be familiar with some of these words. Peptidoglycan, the sugar, and peptides. So that's what bacterial cell walls are made of. Plant cell walls are generally formed only from the polysaccharide cellulose. So the big difference is bacteria have proteins, or maybe not full proteins, but they have amino acids built into their cell wall. And, as it might be, lucky for us, penicillin is able to attack those proteins. Kind of see the images here. So the antibiotic penicillin stops those proteins from linking together. So first of all, hopefully you guys recognize these hexagonal pink and purple shapes. These represent polysaccharides. The linked circles are the amino acids. So here are the again, I'm not sure if they're full proteins or not, but certainly chains of amino acids. And then this green S is a protein. You can kind of see it here. It kind of hooks around this end of the amino acid and that end of the amino acid, and kind of holds the wall together, holds the different pieces of the wall together. And what penicillin does is it targets this particular protein that holds the bacteria cell wall together. And without that protein, penicillin's blocking it from doing its job. Without that protein, the wall isn't held together, and so in essence, it falls apart, it breaks. Now the bacterial cell is osmotically unstable. See that word here? We talked about osmosis a few days ago, last unit. That cell in a hypotonic environment is no longer protected from getting too large. The cell wall is weak. The cell wall has holes in it now. It's not held together. So when that cell's in a hypotonic environment, it's going to swell up with water, and it's going to explode. It's going to pop, and voila, you've killed the bacteria. And that's how penicillin works. Penicillin there, that blue shape blocking the green protein from holding together the wall. Okay, back to our different parts of bacteria. They aren't all bad. In fact, 99% of them, 99.99% of them, we really hope penicillin doesn't do that to them, because we like those guys. Bacteria are actually very important in the world. So back to bacteria. Next up, we're looking at ribosomes. All these little small blue dots are representing ribosomes. And ribosomes are an organelle that have RNA and protein. They're made up of a couple of RNA strands and some proteins. And this is the site of translation. Translation is where protein synthesis happens. It's where we link together amino acids to make proteins. Every cell on Earth has ribosomes. You have ribosomes in your cells. I have ribosomes in my cells. Your dog has ribosomes in its cells. Every cell has ribosomes. It's an evolutionary link to the very first cells on Earth. Typically, I know we've got a lot of blue dots here, but it's not nearly enough. Typically, a bacterial cell will have in the neighborhood of 30,000 ribosomes inside of it at any given time. Making proteins is a really, really important job for the cell. Next up, we're taking a look at the flagellum, which is singular, plural is flagella. It comes from the Latin word, which means whip. And it's this long tail-like structure that you see here. That's the flagellum. Its main purpose in most bacteria is locomotion. So it moves like a tail swimming through water. Generally, those are made of proteins. Bacteria can have numerous numbers of flagellum. So you see this one here has just one, this one has two flagella, this one has several, and this one looks hairy. It's got so many. So the number of flagella all depends on what species of bacteria it is. Flagella are not unique to bacteria. For instance, mammal sperm have a flagellum. and that's how they are able to move. So it's not unique just to prokaryotic cells. Our next set of notes should be on eukaryotic cells. And one of the big differences, yes, they have a nucleus. They also have other membrane-bound organelles. How do they get those membrane-bound organelles? Well, the thought is what's called the endosymbiotic theory. And the idea is that one bacteria cell, this would have been billions and billions of years ago, one bacteria cell would have swallowed up, engulfed another bacteria cell, and then they would have lived together in a beneficial relationship like this. The little tiny one would have gone to work for the bigger blue one. So the endo part of the endosymbiotic theory refers to one cell ingesting another one. You guys remember this term from last chapter? Endocytosis. So that's the endo part of the endosymbiotic theory. Then the symbiotic part refers to the two cells then working together. That's what we know as symbiosis. Both cells benefited from this relationship, so it's the endosymbiotic relationship. Some pieces of evidence that we have found that support the endosymbiotic theory, that explains how eukaryotic organisms got their organelles. First of all, many organelles have a double phospholipid bilayer. So not just one phospholipid bilayer, but two. And again, I'm not just talking about phospholipid heads and tails. Here's one layer. It'd be a second layer in addition to that. Why is that the case? Well, take a look. If endosymbiosis is happening, here's the endosymbiosis. The one cell's being eaten by another one. This first bacterium has its own phospholipid membrane. The second bacteria that swallows it also has its own membrane. So guess what? Now we have a double membrane. We have the cell inside of a cell. The host cell has its own membrane there, and the cell that was swallowed has its own membrane. That leads to a double membrane. And when we get to a lot of the organelles, we'll notice that they have two membranes. This is probably why. The mitochondria and chloroplast, both double membrane bound organelles, are both roughly the size of bacteria cells. So we talked about eukaryotic cells are 10 to 100 times larger than a bacteria cell. Well, some of their parts, mitochondria and chloroplast, are about the same size as a bacteria cell. Here's another very interesting one. Mitochondria and chloroplast each have their own DNA. And look at that. It's also circular DNA, very much like bacteria DNA. Very interesting. And then finally, mitochondria and chloroplast also reproduce themselves. They grow, they elongate, and then they split in two. What does that look like? There's a mitochondria growing, getting longer, and then splitting in half. And that's how our cells replicate mitochondria, and plant cells replicate mitochondria and chloroplasts. As they grow, they get longer, and they split in two. Should look a lot like binary fission. We discussed it yesterday, and then it's there. That's how bacteria reproduce. They grow, they get longer, they split in two. So definitely some very serious evidence for the endosymbiotic theory. I'll leave you there, and we'll pick it up again next week.